Dr. Becky Scalzo. She is a CSU grad, so this has been awesome to have her here. I'm not the person to give her an introduction. Instead, I'm going to have Dr. Chris Bell, who was her supervisor, introduce her formally. So.
That was it.
More time.
So first, I think we should acknowledge that this semester's seminar series is brought to us by the letter L. Let's thank Drs. Lodge and Lodar. I think I'm also supposed to tell you that it's an action-packed seminar series, and next week there is a metabolic legend coming. Sam Klein will be here to talk about metabolism, so he's staying well within his comfort zone. But this week we have a legend in the making. If I may paraphrase a fairly popular book. Rejoice, let us kill and feast on the fattened calf, for the prodigal daughter, she is returned. Of course, in this instance, the fattened calf is obese and probably has diabetes, but that's okay. The prodigal daughter, on the other hand, is alluded to by Dr. Mark, is of course Becky. Despite spending over 4 years in my lab, Becky was able to secure an excellent postdoctoral training experience down at Anschutz with Janusz. I say despite, acknowledgement must be conferred to the co-mentoring and academic repair work of Dr. Hamilton, who also contributed. Upon arrival in Denver, Becky was immediately appointed to the presidential election campaign that later on celebrated the victory of Dr. Roosh as she was appointed head honcho of the American Diabetes Association. And as a consequence, Becky is now available for any presidential campaign that's coming up in the near future. Less than 1 year out of her postdoc, Becky already has 18 scientific publications, 9 of which is the first author. She has a ludicrous amount of money to support her work as an independent investigator provided by the VA. But, importantly, in addition to being a rock star scientist, Becky is a wife, a mom to 2 daughters, a slave to the needs of 2 nerdy, hyper-energetic dogs. She is a proficient baker, an inefficient runner, a frustrated guitarist. She claims that she can knock out a tune on the piano. She reads fiction. She vacations in Mexico. She is a connoisseur of cheap boxed wine.
No! That far.
In short, she is the perfect example to us all of a successful career scientist who has a work-life balance that could be accused of almost being healthy. Let's give her a healthy welcome.
It has been nice to be giving talks independent of Chris Bell because I don't have as much pressure to follow that entertainment value. So that was a special treat. Thanks. I am appreciative of the invitation to be here today. It was a lot of fun to have lunch with the students. You guys have a much nicer setup than when I was here, and there were Jimmy Johns in the conference room. And also it's nice to catch up with faculty and get an idea of what's going on here in the department. I'm here today to talk to you about my research program that's focused on the interplay between type 2 diabetes and estrogen syndrome. So I work down at the medical center within a large collaborative group that's focused on the sex differences in type 2 diabetes across the lifespan. So we study everything from the pediatric population all the way through older adults. And my particular piece of this puzzle is focused on premenopausal women and understanding the burden of the disease in this population. So I have a graphical outline to give you an idea of where I'm going to go with my talk today. Or you can consider this what stands between you and Road 34. So first, I will give a brief overview of type 2 diabetes, what we know to be the sex differences between premenopausal women and their age-matched male peers, and the data I generated during my postdoc to support the hypothesis that type 2 diabetes disrupts the beneficial effects of estrogen in the skeletal muscle. I'll culminate with my plans for the future on this part of my research program, and then I'll switch gears and talk about what happens to type 2 diabetes risk when estrogen signaling is impaired in breast cancer therapies. To talk about how type 2 diabetes risk is greater in a specific population of breast cancer survivors, and what work we've done to try and characterize why that excess risk exists. So to start, type 2 diabetes is a chronic disease that's characterized by hyperglycemia that results from tissues losing their sensitivity to insulin. This hyperglycemia and also the results of hyperinsulinemia wreak havoc on pretty much every organ and system in the body. Type 2 diabetes is something we should all be concerned about because one, it is very common. About 10% of the United States population has type 2 diabetes, and another 80+ million individuals are considered to be pre-diabetic. Two, it's very costly. The current estimates for the direct and indirect costs associated with treating type 2 diabetes exceed $300 billion annually. And three, type 2 diabetes is associated with several devastating comorbidities. The one we are particularly focused on is cardiovascular disease. And that is because most people with type 2 diabetes will die from a cardiovascular event. So this is something that we keep in mind with all of the clinical studies that we design. However, it is not feasible for us to use a cardiovascular event as a primary outcome in our studies. So instead, we use cardiorespiratory fitness. And that's because fitness is one of the strongest predictors of all-cause and cardiovascular mortality. So these data are relatively old, but I continue to use them because I really like the simplicity of the graph. So the investigators here recruited a large cohort of men. Data like these have been repeated with women. And at the beginning of the study, they measured fitness in all of the individuals. And they segregated these individuals by those measures. So the people who started the study with what was considered to be high fitness are represented by the solid line. And those that started the study with relatively low fitness are represented by the dashed line. And you can see years of follow-up here on the x-axis. As study follow-up progressed, the people who started the study with greater fitness were more likely to be alive as the years went on. However, the people who started the study with lower fitness were more likely to have suffered premature mortality. And this is an important concept when we're talking about people with type 2 diabetes because these individuals have significantly lower cardiorespiratory fitness. So this is something our collaborative group's been studying since I was in high school. So these are very old data, but they still ring true. So the way our group studied the impact of diabetes on fitness was to recruit 3 different cohorts to the lab. So the first group was considered normal by BMI. They're indicated here by the term mean. The second group was obese by BMI, but were metabolically healthy. So they had normal hemoglobin A1C and normal fasting glucose, the 2 diagnostic criteria for type 2 diabetes. And then the third group had diagnosed type 2 diabetes. And when they measured fitness, they found that the people with type 2 diabetes had significantly lower fitness than either group of metabolically healthy control individuals. So in people with type 2 diabetes, their lower fitness predicts their greater risk for cardiovascular disease. Now, like I said at the beginning, I'm particularly interested in premenopausal women with type 2 diabetes. And that is because in this population, the relative risk for cardiovascular disease associated with type 2 diabetes is greater than their age-matched male peers. And what's interesting about this relationship is it is predicted by cardiorespiratory fitness. So more contemporary data from our group. So again, we recruited people with type 2 diabetes, indicated by the black bar, and then BMI-matched metabolically healthy controls, indicated by the white bars. When we looked at VO2 peak in the males, we found that there was about 16%, that men with type 2 diabetes had 16% lower fitness than their male peers. However, this gap widens to 24% in women with type 2 diabetes. And currently, we don't understand why the sex difference in the cardiovascular disease risk associated with type 2 diabetes exists. However, because we know that this sex difference in risk is predicted by a difference in cardiorespiratory fitness, my thought process was we could consider the mechanism through which type 2 diabetes lowers fitness and see if we can identify a place that's regulated different between men and women with and without type 2 diabetes. So there are lots of reasons why people with type 2 diabetes have lower fitness. To hit the high points, the first is their insulin resistance. Like I said, that's a characteristic of the disease. Insulin resistance limits how well substrate can be trafficked into skeletal muscle for exercise or for function. The second is cardiovascular function. So before a person with type 2 diabetes is diagnosed with overt cardiovascular disease, they have changes in their cardiovascular system that limit their ability to exercise. And the third mechanism is skeletal muscle mitochondria. So the organelle responsible for taking oxygen and substrate and using those items to generate ATP through oxidative metabolism. And skeletal muscle mitochondria is where I'm going to focus today. And that's where I focus most of my project, partly out of convenience. So when I joined the lab as a postdoc, we were in the process of profiling skeletal muscle mitochondria in people with and without diabetes in quite a bit of detail. So it was something I was already learning quite a bit about. And the other reason is, as we were going through the data, and as I'll show you, this is a factor where we notice that men and women with and without type 2 diabetes appear to regulate differently. So skeletal muscle mitochondria look different in men and women with and without type 2 diabetes. But first, I'll show you what I mean by our lab is deeply enriched in profiling mitochondria. So when we look at skeletal muscle mitochondria, we do it from both a function and a content standpoint. So our first step is to look at in vivo mitochondrial function. And we do this through magnetic resonance spectroscopy. So the way this works is a participant slides into a magnet, does an acute bout of single leg exercise. And then as they recover inside that magnet from that single leg bout of exercise, we can track phosphorus peaks to get measures of synthesis rates associated with mitochondrial function. So here on the top part of this graph, we have one of those measures, which is the synthesis rate for PCR. And on the bottom, you have the maximal synthesis rate for ATP. And you can see that these assessments of mitochondrial function are lower in people with diabetes compared with their BMI matched peers. We also will profile mitochondria from a skeletal muscle sample. So the way we do this, so we will look at respiration sometimes, just like is done here in the Department of the Ouroboros, but we'll also try to characterize how much mitochondria is in that muscle. And we do this from using a composite of measures to be able to draw a conclusion about the mitochondrial content. So we do something similar to this graph. So the top right or left-hand corner, we have PGC1 alpha, which is considered the master regulator of mitochondrial glycolysis. In the middle here is BDAC, which is a mitochondrial membrane protein. And then the far right is citric synthase activity, which is an enzymatic step in the citric acid cycle. And then across the bottom here, we will profile complexes of the electron transport chain by using Western blot. So that's the protein content specifically of those complexes. So when we did that in this study, we were expecting to see that people with diabetes have less skeletal muscle mitochondria. And that's not exactly what we found. Across the board, the black bars look lower than the white bars, which brought us to conclude that these mitochondria look puny. But the only place they achieved statistical significance was for citric synthase activity, So this is just an example of how we profile mitochondria. Now these data are collapsed, men and women. This study wasn't powered or planned to look at sex differences within type 2 diabetes. But as we were getting ready to go with the next study, we decided to change our power strategy and to make sure we recruited enough men and women so we can make comments on the differences between males and females with and without type 2 diabetes. And that is a study that's still in the process of being analyzed. However, I was responsible for the biochemical analysis of the skeletal muscle, so I had a sneak peek look at our measures of mitochondrial content. And when I looked at the males from that study, I saw absolutely no difference between those with and without type 2 diabetes. However, the story was quite different when I looked at the female participants. So this was a training study. So these graphs are set up a little bit differently. So we have, these are all women, so women with type 2 diabetes and their BMI matched female peers. I should mention, when we study premenopausal women, we always take our staples during the early follicular phase of the menstrual cycle, so we standardize that. Since this is a training study, the baseline data are represented by the white bars, and the post-training measures by the black bars. So we look at our measures of mitochondrial content. We see that for complex 1 and complex 3 electron transport chain, BDAC, this is mitochondrial membrane protein, it's a trypsin-based activity, that women with type 2 diabetes appear to have lower mitochondrial content compared with their female peers. And additionally, we have another study going on right now. We're looking at mitochondrial respiration, so an ex vivo assessment of mitochondrial function. And I took a sneak peek at those data and found too that women with type 2 diabetes appear to have lower mitochondrial respiration. This is specifically for uncoupled respiration with carbohydrate substrates, compared with their non-diabetic female peers. So to review, women with type 2 diabetes have a relatively greater risk for cardiovascular disease, and that is predicted by their relatively lower cardiorespiratory fitness. I was on the path to try and identify something that was different between women without type 2 diabetes that could be predicting that difference in fitness, and it appears that mitochondria might be one of the players in this party. So my next step was to consider why mitochondria were regulated differently in women with and without type 2 diabetes. And my first thought was that it was an estrogen problem. One, because these are premenopausal women and estrogen regulates everything in young women. And two, estrogen is one of the most powerful regulators of skeletal muscle mitochondria. Everyone sitting in this room has 3 estrogen receptors. You have estrogen receptor alpha, estrogen receptor beta, and a G-coupled estrogen receptor. In human skeletal muscle, there's very low expression of estrogen receptor beta, so I'm going to ignore that one today. But the other 2 receptors are present, and specifically for estrogen receptor alpha, there are several ways that estrogen can regulate mitochondrial biogenesis. So estrogen receptor alpha, when it's in the cytoplasm, can have direct or transcriptional regulation of endothelial neptune oxide synthase, PGC1 alpha, so that's master regulator of mitochondrial biogenesis, and actually mitochondrial content. So there's estrogen response elements on several of the electron transport chain complexes. Estrogen receptor alpha can also be bound to the cell membrane, and when it's bound there and estrogen binds this receptor, it will signal through PI3 kinase and AKT to activate this metabolically sensitive pathway, also culminating in mitochondrial biogenesis. The G-coupled protein estrogen receptor is always docked on the membrane, and it signals through cyclic AMP to activate AMP kinase, which also will signal to mitochondrial biogenesis. So those pathways are specific to making more mitochondria, but estrogen will also regulate mitochondrial function by altering the mitochondrial membrane potential and mitochondrial oxidant production. So estrogen is a very powerful regulator of skeletal muscle mitochondria. And there is precedent from other disease models that estrogen-mediated support of skeletal or mitochondria is disrupted. The 2 examples that I'm aware of, one is from neurodegenerative disorders. So in the brain, when those disease processes are present, estrogen will not signal to make more brain mitochondria, limiting the function in that tissue. And also, one of our collaborators down at the medical center is studying how type 1 diabetes disrupts the estrogen signaling in the vasculature to drive the cardiovascular phenotype in people with that disease. So there is good reason to think that diabetes could be disrupting estrogen signaling based on those other disease models. But also, when I looked at the signaling diagram, I noticed that there were two obvious places that we know that diabetes alters function. So one is at this PI3 kinase AKT part of the pathway. So PI3 kinase and AKT are part of classic estrogen, or excuse me, hormones are classic insulin signaling, which is impaired in people with type 2 diabetes. If diabetes is also altering how these signaling molecules interacted, it could alter the beneficial effect of estrogen signaling through estrogen receptor alpha. And diabetes alters calcium handling and how CAM kinase activates its downstream targets. So in theory, type 2 diabetes could alter that part of the pathway as well, changing how AMP kinase is activated. Currently, I'm unaware of how diabetes would be directly impacted with the directly genomic impacts of estrogen, although as you'll see as I get towards the end, I'm going to try and figure that out. So I have data from women, humans, with and without diabetes that suggests that estrogen, or that diabetes is modifying their muscle mitochondria profile. So I decided I wanted to continue to pursue this line of questioning, but I needed to switch to an animal model. So as I move forward, I would be able to manipulate particular pieces of the puzzle more eloquently. So my first step was actually a step backwards. Because I learned as a postdoc, one of the things I was down at the medical center to learn was to work within animal models of disease. And I learned that unfortunately, animal models do not always behave the same way as humans. So my first step was to ensure that there was, that a sex difference persisted in a rodent model in a skeletal muscle mitochondria. So I took male and female waste-star rats and I fed them either a standard chow diet or high-fat diet to push a diabetic phenotype. And then at sacrifice, I harvested their skeletal muscle and I followed the signaling pathway associated with estrogen-mediated support of mitochondria. So looking first at AMPK, which can signal and activate ENOS, then PGC1 alpha, and then again looking at mitochondrial content by Western blot. So for these graphs, the males are on the left and the females are on the right. The chow or standard diet are represented here by the white bars, and the high-fat diet or metabolic challenge is seen in the black bars. And you see that for phosphorylated AMPK and the ratio of AMPK to total input, right, there's a significant interaction between sex and diet. So in response to this metabolic challenge, the males have activated AMPK, and this response is absent in the female animals. The next step was to look at ENOS. So again, phosphorylated ENOS and now total ENOS are greater in the male animals in response to this high-fat diet or metabolic challenge, without a response in the female animals. And PGC1 alpha, down here at the bottom, followed a similar pattern. We looked at mitochondrial content by Western blot. So these graphs are set up with the males in the blue and the females in the pink. The darker bars represent the high-fat diet challenge. You see what would be expected based on this signaling cascade, that complex 1 and complex 4 protein content are greater in the male animals, and that's absent in the females. So like I said, this study was meant primarily to be a quick screen to make sure that female rodents were going to behave the way I would hypothesize for the humans before I moved forward with further investigations in a rodent model. So to better detail the impacts of diabetes on estrogen signaling, I moved to an all-female model, and I also moved to a mouse model, and that was primarily for logistical considerations. So in this model, mice were fed either a low-fat diet or a high-fat diet. And with all of our mice, we keep them in a thermoneutral environment to drive or exacerbate the impact of the high-fat diet on the development of obesity and diabetes. They don't quite look this fat, but they do get nice and chubby. At 10 weeks of age, all the animals were over-optimized, so we removed all endogenous estrogen in these animals, and then half of the animals received estrogen and back. So each group, there was a group that had estrogen and back. And then again, harvested skeletal muscle and identified how diabetes and estrogen interacted to manipulate the same signaling pathway. So for these graphs, the low-fat diet animals are here on the left, high-fat diet animals on the right, over-optimized alone in the white bars, and over-optimized plus estrogen in the black bars. And so for AMPK, we saw no effect of diet or estrogen. And then we moved to activated enos, so this is phosphorylated total enos. And in the low-fat or the control animals, so back up, there's a significant interaction between diet and estrogen. What we see in these control or low-fat animals here is what I would consider to be the expected contribution with estrogen. So greater enos activation with estrogen, and this response is absent in the high-fat diet animals. Next, we looked at PGC1 alpha, where we saw a similar pattern if these data did not achieve statistical significance. And then I threw in one kind of oddball measure. So OPA1 is a protein that regulates mitochondrial fusion. So the process through which mitochondria come together, in theory, joining more connected and effective networks. I threw OPA1 into my analysis because at the time I was doing this, there were some really great papers coming out linking estrogen signaling to OPA1 and mitochondrial fusion. And I thought it would be a good idea to be able to link to that contemporary literature. So when I looked at OPA1, I again found a significant interaction for diet and estrogen. So greater OPA1 with estrogen in the low-fat animals and no effect of estrogen with high-fat diet. So these data together initially led me to think that I was on the right path, that diabetes and high-fat diet are interfering with estrogen signaling to skeletal muscle, to skeletal muscle mitochondria. But someone pointed out that I didn't know if that was because the receptor changed or the downstream signaling changed. So to address that, I looked at the gene expression of estrogen receptor alpha, that most prominent estrogen receptor for signaling to mitochondria, and found no effect of diet or estrogen on its expression, suggesting that these changes could be because of downstream signaling changes. So I took these data in addition to the story I've been weaving about lower cardiorespiratory fitness in women with type 2 diabetes. In humans, they've got lower mitochondria compared with their female peers. And I wove it into a story to support my career development award, which is aimed at testing the very general hypothesis that type 2 diabetes interrupts the benefits of estrogen on skeletal muscle mitochondria. So this is an award I've had for 6 months, so I have absolutely no data from it. But I did bring my the scheme of my research plan to give you an idea of what I plan to do. So I acknowledge that the data I built through a series of small pilot grant-funded studies don't wholly characterize the impact of diabetes on estrogen. So I plan to correct that. So specific A1 is all in a primary cell culture model where I plan to use normal and conditioned media to replicate type 2 diabetes. And estrogen and estrogen receptor antagonists to determine the impact of estrogen through both genomic and non-genomic signaling pathways on these targets associated with mitochondrial content and function, and also determine the role of diabetes or the interference that diabetes plays in this process. I also, in specific A2, will use a whole animal model to better characterize the phenotype of the manipulation of diabetes and estrogen. So using a similar setup to that mouse model I showed you, but do a deeper dive in phenotyping these animals. Also understand how these changes impact their functional capacity so we can measure exercise capacity in the animals. And also pull out their muscle for assessments of mitochondrial biogenesis using deuterium oxide and also mitochondrial respiration using oral pills. So my goal with all these projects is to, in a couple years, be able to identify skeletal muscle targets that can be used specifically to treat women with type 2 diabetes and help relieve that relative decline that they experience in cardiorespiratory fitness. But like I said, I've got another 4.5 years on that award, and I'm going to need a lot of that time to be able to address this goal. So that's the first part of my research study, and that really represents a lot of the data I collected in the last 1.5 years of my postdoc before I transitioned to a faculty member. But as I was doing that, I met a woman who's a mammary gland biologist, and we just happened to be sitting together at a luncheon, and she was telling me that she just stumbled upon this idea that one breast cancer treatment used specifically in young women increases the risk for developing type 2 diabetes. And she, as a mammary gland biologist, sorry, I can't say that, doesn't really know anything about type 2 diabetes, but has a lot of the skill set necessary to study this problem. Asked if I wanted to partner with her in trying to figure out why these young women with breast cancer are at greater risk for developing type 2 diabetes. And I said, of course. One, because I was junior faculty and I need collaborators. And two, I thought this would be a really good way to serve the other side of my research program and learn more about estrogen signaling from someone who does this for a living. So to be able to do this, I had to first learn about breast cancer myself. So every year there are about 200,000 new cases of breast cancer in the United States, and 75% of these are estrogen receptor positive tumors. Tamoxifen is a selective estrogen receptor modulator, and in the breast it behaves as an estrogen receptor antagonist. Excuse me. Because of that, tamoxifen is the first-line drug choice for premenopausal women with these hormone receptor positive tumors. Tamoxifen is prescribed from the day a young woman is diagnosed with one of these hormone receptor positive tumors, and she will take that drug until she transitions to menopause. On average, this is about 5 years. And the reason tamoxifen is used so much is it alone can reduce the reoccurrence of breast cancer in young women up to 50%. However, both prospective and retrospective epidemiological data suggest that tamoxifen use increases the risk of type 2 diabetes. What's interesting is that the normal weight women are treated with tamoxifen. They do not develop insulin resistance. However, when overweight and obese women are treated with tamoxifen, they do. And so when they've looked at rates of type 2 diabetes in these women, their risk exceeds what is seen in their cancer-free peers. So when they match for age and for BMI, something about the combination of tamoxifen and obesity is driving an exaggerated type 2 diabetes risk. From clinical data, we know that breast cancer survivors treated with tamoxifen develop hepatosteatosis. And this excess lipid deposition in the liver could be the obvious cause for their elevated type 2 diabetes risk. However, I'm an integrative physiologist, and I appreciate the contributions of other tissues to the development of type 2 diabetes. And if I'm honest, the liver scares me. And what we don't know is how tamoxifen impacts these other metabolically relevant tissues. So our goal together, my collaborator and I, was to first develop a clinically relevant mouse model to be able to study the interaction of obesity and tamoxifen. And two, to address the hypothesis that the detrimental impact of tamoxifen on insulin sensitivity is mediated through changes in the liver and the skeletal muscle. So we set up our model similar to my diabetes and estrogen model. So female mice that were fed either a low-fat diet or a high-fat diet. Again, they were kept in the thermoneutral conditions to exacerbate the impact of the diet. And then all the animals were once again over-optimized at 10 weeks of age. Half the animals received estrogen add-back, and the other half received estrogen plus tamoxifen. So as our first goal was to characterize the animals. Well, let me back up. So on these graphs, the low-fat diet animals are here on the left side, high-fat diet animals here on the right side. Estrogen alone are represented here by the white bars, estrogen plus tamoxifen represented by the black bars. So we looked at these animals both for body mass and for body fat percentage. There was a significant interaction between diet and tamoxifen use. So tamoxifen exaggerated the impact of the high-fat diet on these two variables. Because this was our first attempt at using this model, we used both the glucose tolerance test and fasting glucose and insulin to screen these animals for both glucose tolerance and insulin sensitivity. So here we have glucose area under the curve from the glucose tolerance test. And we saw that the high-fat diet animals had lower glucose tolerance indicated by their greater glucose area under the curve. However, tamoxifen did not impact that variable. When we looked at fasting glucose, however, we once again saw this significant interaction for diet and tamoxifen. So the high-fat diet animals treated with tamoxifen were hyperglycemic. We looked at insulin next. We found an effect of both starting affected tamoxifen and an effect of the high-fat diet. However, there's no interaction of these two variables for insulin. And finally, we calculated a home IR. So we estimated insulin resistance in these animals. And we found, once again, this significant interaction for diet and tamoxifen. So from a whole animal perspective, we were happy with our model development. The high-fat diet tamoxifen seemed to exaggerate the impact of the high-fat diet. These animals were hyperglycemic and they were insulin resistant. So our next step was to characterize the liver, because this is the one piece of data we have from the clinical patients, or the breast cancer survivors. So we looked first at the liver histologically. Hopefully you can observe that high-fat diet animals treated with tamoxifen had a lot of lipid in their liver. So all of these empty white spaces here are lipid accumulated in the liver. We quantified these data by looking at liver triglyceride content. So when we did this, we found again a significant interaction between diet and tamoxifen use. So the livers looked fattier, and when we quantified it, they were fattier. And we were curious, as these are the two pieces of data that relate to the clinical population, the liver fat and insulin resistance. How much of liver triglycerides accounted for the insulin resistance we're seeing in our model? So we ran a correlation between these two variables and found that while it was positive and significant, when we looked at the R-square value, it appears that the liver triglyceride content is only accounting for about 40% of the insulin resistance in these animals, which I was happy about because that gave us permission to look at other tissues. The first place I went was skeletal muscle, because as you can tell from the first part of my talk, that's where I'm most comfortable operating. And again, I chose to look at the mitochondria profile. One, because changes in skeletal muscle mitochondria can have an obvious effect on whole animal insulin sensitivity. And two, if tamoxifen is targeting the estrogen receptor in the skeletal muscle, it's possible that there'd be changes in this pathway, like I saw in my other animal model. So we looked at the data. We found greater phosphorylation of AMPK with tamoxifen, and lower phosphorylation of AMPK with the high-fat diet, and lower phosphorylation of ENOS with the high-fat diet. But no clear signal that obesity and tamoxifen are interacting negatively on this pathway. Also looked at mitochondrial content by assessing the complexes by Western blot. And some could claim that there may be a couple of differences, specifically in complex 2 and complex 5 in the high-fat diet animals treated with tamoxifen, but those p-values didn't attain statistical significance. And the final piece in the mitochondrial puzzle was looking at certificate activity, where we found greater activity with tamoxifen, but no effect. So these data sadly led me to conclude that there's nothing shaping in my favor that way. It doesn't appear that tamoxifen and obesity are negatively impacting the muscle mitochondria. But because of what we saw in the liver, we were also curious just what these skeletal muscle samples look like histologically. And when we did that, we noted that, same position, high-fat diet animals treated with tamoxifen have these large lipid droplets in between the muscle fibers. And their muscles as a whole look like a marbled steak. I don't know if you can appreciate that in there, but their coloring looks different because there's so much white in those muscle fibers. This is not something we are anticipating finding, so our next study is designed to both quantify and qualify what this fat is in the muscle, because a change, or ectopic lipid deposition, specifically if it's some of the more reactive species of lipids, could be driving part of the insulin resistance we see in these animals. So our final place to look was adipose tissue. tissue. And we specifically were looking at the process through which resident progenitor cells in the adipose transition to pre-adipocytes, and then as nutrients are available, fill and become mature adipocytes. And we are interested in this pathway because it is facilitated by estrogen signaling. And it is plausible that tamoxifen, inhibiting the estrogen receptor, could impair this transition between progenitor cells and pre-adipocytes. And if that were happening, what we would have expected to find were a larger number of hypertrophic, overfilled adipocytes, especially in the case of overnutrition or high-fat diet. So first, we looked at the progenitor cell population in this adipose tissue and found that the high-fat diet animals treated with tamoxifen had fewer progenitor cells in their adipose tissue, and this corresponded to a greater number of large adipocytes. So for this graph here, this is the adipocyte diameter on the x-axis and percent of total. So a relative number of how many of each adipocyte type we have. And you see that for the high-fat diet fed animals treated with tamoxifen, that the profile looks skewed to the right, or skewed to larger at the adipocyte diameter. We also quantified the average diameter of the adipocytes from each animal group and found, again, a significant interaction between tamoxifen and the high-fat diet. So a greater number of larger adipocytes in those animals. So putting all of those data together, this is the working model we've come up with to explain why young women treated with tamoxifen are at increased risk for developing type 2 diabetes. So tamoxifen impairs their ability to expand their adipose tissue. So when there's extra nutrients available in the context of obesity, this causes an overfilling of adipocytes, which leads to lipid spillover and ectopic lipid deposition in both the liver and the skeletal muscle. We have, within this study, we've tried to go both directions on the translational spectrum of our future study designs. One aspect of our next study is aimed at describing these progenitor cells and pre-adipocytes better and using bioinformatic methods to better understand what those populations look like in the context of obesity and tamoxifen treatment. And our other study is more in my wheelhouse, trying to identify a treatment that can be used in conjunction with tamoxifen to reduce the risk of type 2 diabetes development in these women. So we took mice that had a high-fat diet and treated with estrogen and tamoxifen, and we put one group through aerobic exercise training, put another group on metformin in addition to tamoxifen, and a third group on pioglitazone, which is a TZD that specifically targets lipid accumulation in the liver. And we're currently working through those data to see if there's a clinical outcome we can grab from this preclinical study to maybe push towards a treatment option for women, young women with breast cancer. So that wraps up the two studies I have going are arms of my research program that are dealing with the cyclical nature between type 2 diabetes and estrogen signaling. So what I hope I've shown you is that it appears that type 2 diabetes is interfering with the benefits of estrogen and skeletal muscle, and that I have plans to characterize that further with one of my current awards. And also that if you alter estrogen signaling in the context of breast cancer therapy, you can drive type 2 diabetes development within the context of young women. So with that, I want to thank my collaborators and funding sources. I'd like to specifically highlight my postdoctoral mentor who supported me through a lot of the preliminary data generation, and also my primary collaborator for the breast cancer studies is Dr. Elizabeth Fuller. So with that, I'm happily take questions. Yep.
So there's kind of layers to this question. And I know you can only do some other things at once, so I could just do it with low priority. So you're really interested in receptor dependent effects of estrogen.
Yeah.
So two kind of layers of questions there are, can consider any of the receptor-independent effects of estrogen. And the other layer is mostly less stubborn also. I think lots of different cell types have ER receptors, both on the passive membrane and the nuclear membrane. You didn't have any differences in estrogen receptor protein expression. But what do you know about the distribution of those receptors and if that might influence your genomic immunology?
So to answer your first question about the difference between estrogen acting through its receptor and estrogen acting independent of its receptor, that's part of my plan moving forward. So one of the many arms of that, those primary cell culture experiments is to block all estrogen receptors and then put estrogen there and see what happens. To answer your second question, yes, I looked at estrogen receptor alpha. Actually, I looked at gene expression. I learned how to do PCR. But I looked at it in a whole tissue assay, not location specific. I kind of considered looking at it location specific, but I definitely could. But I'm gonna have plenty of tissue available, so I'll do that.
I don't even know if it's a good idea, but
All
the
in.
muscle's so complicated since there's so many nuclei and non-nuclear vein, it takes time and different parts of the muscle fiber.
Right. And I also have plans to use inhibitors against several signaling pathways associated with the direct or the membrane-bound receptor signaling to try and knock that all out and see what happens. And I floated the idea of trying to do using CHIP to figure out if that's what estrogen binding to. I kind of do a lot of things. I've learned a lot more since that grant was awarded about estrogen signaling and how to measure it, and I have a lot of opportunity moving forward. Yeah.
So I guess big picture question, is the prevalence or incidence of type 2 diabetes greater in women compared to men? And then a follow-up question to that is, you're sort of stating that diabetes is reducing the positive benefit of estrogen, but aren't estrogen concentrations going up with obesity? So can you kind of talk about if you've measured that or how that influences your
Right,
results?
so to make sure I understand how many women have type 2 diabetes relatively, and how to handle greater potential estrogen concentrations in obese women. So to answer your first question, in the adult population, it's equal between males and females with type 2 diabetes. To be fair, it's more prevalent in an older population. However, as I mentioned at the onset, we study the pediatric population with type 2 diabetes, and it's sad that that even exists. In the pediatric population, there's more girls and boys that have type 2 diabetes. So if you consider they are aging into the adult population as we go. And also, in people that are considered to have pre-diabetes, that's more female than male. So I definitely did hedge my bets on the problem is coming, that there are going to be a large number of young women with type 2 diabetes. So does that address your first question? Okay. And then the second question is about greater estrogen circulating in obese women. So you're right, oftentimes women, obese women have greater concentrations of estrogen, and that's because adipose tissue has aromatase, and that will take testosterone and generate estrogen from that. It is a possibility I've considered, except for we see those differences in mitochondria in the women, the females, the humans. And those are BMI matched, and they're, we don't purposefully match for adiposity or body fat percentage, but they end up being the same. So in theory, they have the same amount of adipose tissue that could be generating excess estrogen. We have just recently added it to our orders to start measuring estrogen in our women. It's just been an issue. We take tissue samples at so many different points, and we usually do our blood test as part of the screening visit, which is not cycle dependent, because there's only so many things we can do that are cycle dependent in these women. get everything scheduled with our clinical and translational research center. So we've moved to trying to, we've thought about measuring estrogen when they come in for their biopsy and the clamp. However, we do that in the early follicular phase. So we're trying to catch people at pretty bottom levels of estrogen then. So I'm not sure what we learned from it, but I recognize your point.
Your early slide showed the influence of cardiorespiratory fitness on prevalence of type 2 diabetes. And your primary outcome is VO2 max. But VO2 max is thought to be limited primarily by cardiac output. So why are you focusing on muscle and not cardiovascular?
Well, the other piece of the very large puzzle from our group is very eloquently characterized in the cardiovascular system. So we're using 4D MRIs with the heart and all kinds of fancy vascular measures, but that is my postdoc mentor space. And while we will continue to work together, that's her piece of the puzzle that I get to play with. But I'm interested, well, one, that's her piece of the puzzle that I get to play with. And two, all those things downstream feed to the skeletal muscle. You still have to have functional skeletal muscle for the engine to go. Looks like you want to follow up. Right.
Yeah, thanks. Just wanted to follow up a little bit with Chris was saying there. Did you measure cardiorespiratory fitness in your animal experiments?
So I am doing that in the rats that I start next week. I was telling Dr. Bell that I was able to assemble my own metabolic rat treadmill with tools all by myself. So I don't have that measure yet. In a subset of the mice that I looked at, I did exercise train them and found that the high-fat diet animals, according to those data, the high-fat diet animals with estrogen didn't respond. There was a significant interaction for the data, but why I don't trust them and why I didn't present them is the high-fat diet animals without estrogen had this massive change in fitness with the exercise training program, and that just doesn't make any sense to me. So I want to repeat that before I'm willing to share that a little bit more publicly. I guess I just did that.
I'm just wondering if it's going to mirror the human condition.
Right.
You know, so you did
so
the
that's
experiments with the muscle, but I'm wondering bigger picture if you're going to bank this all on cardiorespiratory fitness.
Right. No, your
Doesn't
point
follow.
is well taken. And as I was explaining to Dr. Hamilton earlier today, I designed these rat studies just like I would have a human study, which is a little bit goofy. So what I'm going to do to these rats is I'm going to VO2 max test each of them, and then I'm going to do a submax test and collect metabolic data so I can assess changes in substrate utilization. Or differences in substrate utilization at submax exercise intensity, because that would allow for some comments on skeletal muscle metabolism, and actually do run the fatigue test on them, which is a more classic measure of functional capacity in a rat. So I do plan to collect those data, I just have not yet.
Can I ask another question real quick? So the second question I had is there a disturbance within the cell that's already occurring that's sort of independent of estrogen signaling? So is the problem already there and estrogen's trying to do its job and the problem's already in the cell, or is it once estrogen binds the receptor and stimulates whatever this cascade of event is the problem at that point? Like, where is this happening in the cell? Because it seems like it may just be abnormal cell function that, yes, it's dependent on the estrogen receptor signaling cascade, but I'm trying to just figure out, because you have receptor expression, we don't know about the concentrations, we don't know about the binding of it, the sensitivity of it, so I'm just trying to figure out in that cascade of events where the problem lies.
So I believe the problem is already in the cell. The diabetes has made that cell sick, and that is impairing estrogen's ability to do what it would normally do. That's purely based on the idea that, at least for membrane-bound estrogen receptor signaling, you run through enough pathways that diabetes disrupts. I can't answer that question further until I do some of my experiments, and that's what I'm trying to figure out. I think that diabetes is too much of a problem, and that that's what's changing the cellular environment, and not a defect of estrogen itself. Does that answer your question?
Oh, a little bit, yeah. Thanks.
Yes, ma'am.
Have you ever looked at whether this phenomenon is also, because I know in polycystic ovarian syndrome research, there's a very similar phenomenon where most people who are diagnosed with polycystic ovarian syndrome go on to develop prediabetes or type 2 diabetes, and it is also an estrogen-based disease, estrogen and progesterone-based disease, and there's a lot of similarities of the treatment based on what you've been talking about. So have you looked at the comparison of how those two interact, or are they is it the same pathway being affected, or have you ever?
So I've done nothing with polycystic ovarian syndrome, but one of our collaborators, Dr. Melanie Pre-Green, studies specifically, usually, well, she's a pediatric endocrinologist, so she only studies young girls and I guess young women with PCOS. And she is currently doing a lot of the same mitochondrial assessments. At least, I back that up. She is doing the in vivo assessment of mitochondrial function. It's very tricky to be able to get a muscle sample from a pediatric patient. So she's not addressing that question, at least as of right now. So, maybe?
Okay.
I don't know what all it is. Yep.
So the other treatment for premenopausal women with breast cancer that isn't tamoxifen are aromatase inhibitors. And I don't think that women on aromatase inhibitors do have higher risk to diabetes. So I'm suggesting it's not a lack of estrogen problem, it's a tamoxifen problem.
Oh no, it's a specific tamoxifen problem. So to back up, Dr. Braun brought up the other treatment used for these hormone receptor positive tumors. So aromatase is generally not used, at least alone, in premenopausal women, because an aromatase inhibitor will target estrogen that's being generated, like to Dr. Richard's question, in non-ovarian sites. So if you give a premenopausal, so that's why tamoxifen is given to premenopausal women, because instead of trying to block the little trickle of estrogen that's coming out in like a postmenopausal woman or a woman without competent ovaries, you just attack the source, and that's blocking estrogen signaling at the breast. Aromatase inhibitors are used either in combination with something else in premenopausal women, or that's what women who've been on tamoxifen transition to. And that is because they no longer need to worry about the big surge of estrogen from the ovaries, they just need to handle that little trickle that's coming from the adipose tissue. What's interesting about the difference between women on aromatase inhibitors and women on tamoxifen is that aromatase inhibitors drive cardiovascular disease risk. And one of the reasons that they think that's happening is because when they inhibit aromatase in the adipose tissue, they drive up testosterone level. So they end up making those women look like men. So it's kind of pick your poison, which, I mean, well, and it's an age thing. Obviously, a treatment choice is one makes with their physician. But why it's specifically a tamoxifen issue is tamoxifen behaves differently either as an estrogen receptor agonist or antagonist based on what tissue it's active in. And it changes its behavior if estrogen is present. So when there's not, so bone is a great example. When estrogen is present or not present, tamoxifen can act as either an estrogen receptor agonist or an antagonist. So it's a really goofy drug that targets all these different systems and maybe not clear or defined ways yet. Because most people who study tamoxifen have used it without opposing estrogen. They've tried to use it as an experimental model of blocking an estrogen receptor. But that doesn't quite relate to the clinical population, you see what I'm saying? So, it's confusing.
Thank you.
One more question?
Sure. So, interpreting some of your mitochondrial data, you were showing complex protein content relative to alpha tubulin. And then you had
Sometimes we do this with
okay. And then you had citrate synthase activity, which demonstrated that it was lower. The activity was lower.
So you're talking about the people with diabetes.
Yeah, the people with diabetes.
Yep.
And then you also did the same thing with the rats as well. I'm curious as to whether or not this is more of a content issue, because citrate synthase can also be a marker of mitochondrial mass.
So actually, we always include it as in the composite of mitochondrial content, but I don't think it belongs there. The group, one of the European groups walked through all the different indicators of mitochondrial content. I think their point was to detail what you should be normalizing the respiration data to, the mitochondrial DNA, citrate synthase activity, and wet weight. Yep. And they found that citrate synthase activity is not a representative of content. But it's kind of a goofy thing to stick in there because everyone kind of thinks of it as associated with either mitochondrial content or function, but it's also upstream of the electron transport chain. So really what I'm telling you is I agree. It doesn't drive because it's a thing.
So I guess my question, was going to be more so related to whether or not you normalize your activity to be done with, have you done the OTK work then?
We always do the wet weight.
Always do what with muscles?
Mm-hmm.
So have you considered normalizing to measures of mitochondrial content? Is that, because there might
So
be a
the
lower
Western
mitochondrial
blot
content.
assessment?
Yeah.
Yeah, we've started to think about that. John Tifo is doing that in his work, and we always save what comes out of the Ouroboros.
Yeah.
So we could do it, we just haven't taken that step yet.
All right, let's thank Becky for doing that. So I just wanted to follow up before you guys go. Chris mentioned it. So this month is Metabolism Month. So we have Becky this week. Next week we have Sam Klein, who really is an icon in the field of metabolism. He's going to talk to us about normal versus abnormal obesity and the metabolic implications. The week after that, we'll have Dr. Chris Perry from York University in Canada. He is doing all things mitochondria. He touches on muscle dystrophy, cancer chemotherapy. And so he's going to hit on a bunch of different topics, very heavy metabolism. And then finally, we'll have Chris Gentile come in from Food Science and Human Nutrition, talk to us about the gut microbiome and vascular disease. So it's going to be a great month, so stay tuned.